We continue now with our second talk on the digestive system. And in this presentation, we'll consider the reflexive and endocrine control of the GI system. Then we'll talk about, very briefly, the digestion of the major nutrients, carbohydrates, proteins, and fats. And then we'll consider a bit on absorption. Starting first with the neural integration or regulation of the GI system, we think of long reflexes that are coming in from the CNS, short that are integrated into the ENS, and we talked about the anatomy of the enteric nervous system in the first lecture. And of course there's going to be reflexes that also integrate GI peptides, either for local paracrine effects or more remote endocrine effects. Let's first start then with this illustration, kind of a concept map of the integration of the long and short reflexes in the digestive system. So long is shown here and short here, and we're going to look at these in a stepwise fashion. So again, we have our key, the stimulus, the receptor, the integrating center, the efferent pathway, the tissue response, and the red arrow are the short reflexes that we're considering first. So there's going to be local stimuli, such as the acidity, hydrogen ion concentration, stretch due to content, osmolarity, again content, and the products of digestion. So the smaller molecules that are generated through the digestive process can have local stimulatory effects on sensory receptors and neurons nearby. And of course these will project to interneurons. Remember I said there's the submucosal and the myenteric ganglia, as well as the plexuses that we see here. So there's neurons, cell bodies scattered throughout that are not strictly the parasympathetic postganglionic cells. So these are other neuron populations, part of the enteric nervous system or the little brain. So these interneurons, after the sensory neurons and receptors give them information, will contribute to the autonomic components, the myenteric and submucosal plexuses and the ganglia corresponding to the parasympathetic part of this. And of course they target the smooth muscles or the secretory cells, the effectors, and this leads to muscle contraction or relaxation, depending upon whether it's sympathetic or parasympathetic that's activated, right? And the parasympathetic is going to lead to the contraction, and the sympathetic will relax it. Or maybe I've got that backward. Are you correcting me? Double check that, please. The exocrine secretion, of course, the enzymes, the mucus, the acid, and the bicarbonate, and any paracrines. So the neural system can target effector cells that can be endocrine, where they'll either be acting locally as paracrine chemical messengers, or be carried away in the blood for more distant effects or targets. Now we're looking at the gray arrows for the long reflexes. So there's going to be some type of external stimulus, sight, smell, or whatever other sensation there may be. The sensory receptors that pick these up will contribute to the cephalic brain. So these are the higher order neurons in the cerebral cortex that are picking up the information from these ultimately to be integrated into divergent pathways that ultimately will affect the sympathetic and parasympathetic neurons in the spinal cord and the cranial nerve nuclei, of course, for the parasympathetic. Remember, the parasympathetic is craniosacral and it's thoracolumbar for the sympathetic, referring to the anatomical location in the CNS of the preganglionic neurons. Of course, the local stimuli are also contributing to this integration by ascending information. Don't forget about our friend the vagus nerve, a tremendous sensory innervation to the gut, all the way to the transverse colon. And the vagus nerve carries information to the brainstem that is shared with higher centers as well, and these become integrated into the entire output of the autonomic nervous system. And then, of course, you get the effects on the autonomic plexus and ganglia. Postganglionic parasympathetic neurons are in the ganglia, in the GI tract, running parallel, as we said, with the enteric nervous system, or really part of it, but still, we can leave it at that. And then the smooth muscles or secretory cells are the effectors, and we get contributions to the same things. So you know that you can smell the turkey or the pie cooking on Thanksgiving, and the saliva will be secreted into the oral cavity, and stomach might start churning and gurgling and stuff like that. And then finally, looking at the short reflexes and the long reflexes together, we see that there is a convergence onto the same processes. So there can be a cephalic phase that's anticipatory to a meal, and it primes things, if you will, and gets fired up to be coordinated once the local stimuli strengthen and reinforce that. As well, there are reflexes involving the gut peptide signaling systems. So the overview that we see here, there can be a stimuli, the cephalic phase of digestion, which is a feed forward, distension of the gut tube throughout its length, and the acid or the presence of food can be the broad spectrum of stimuli that will act on some type of receptor that will be in the integrating center, in this case the secretory cells of the stomach and the small intestine. So they might be acting as both the receptor and the integrating center, determining the synthesis and secretory rate or secretion rate of the chemical signals, GI peptides, which is the efferent pathway into the interstitial fluid and acting locally or being delivered via the plasma. And the effectors, the target cells, are the smooth muscle in the gut tube, endocrine cells of the stomach, pancreas, and intestine, other endocrine cells, and the nervous system in general. Then the responses are changes in GI motility, release of bile and pancreatic secretions, enzyme acid and bicarbonate synthesis, release, hunger, and satiety, which is one area of great interest to all of us, because if we can understand this better, we can help to regulate eating disorders and the overconsumption of unneeded calories in humans. We're all too aware of that. It's harder to burn more than what we take in. It's a lot easier to take in a heck of a lot more than what we burn. So that's a very active area of research, as well as all the rest, because there are pathologies and developmental aspects that we need to know about in order to understand how things work, as well as to treat any type of clinical condition. So the enteric nervous system shares features with the CNS, reinforcing this idea that it is a separate nervous system in and of itself. It has intrinsic neurons, there's neurotransmitters and neuromodulators, there are supportive cells, there's a diffusion barrier, and there's an integrating center. So it is nearly complete. I refer you to the table in the textbook to study more details on these digestive hormones. I was hoping that the table would be included in this slide, but for the sake of time, we'll just refer you to the outside source to look at more detail. So there's the gastrin family, which includes gastrin, and CCK, which is cholecystokinin. So the cola makes you think of bile. Cyst is the gallbladder, and kinin means to make it move, so the cholecystokinin. Then the secretin family, obviously secretin is going to be targeting some cells to stimulate secretion. Vasoactive intestinal peptide, gastric inhibitory peptide, and glucagon-like peptide 1, which promotes satiety and also stimulates insulin release and inhibits glucagon release in gastric functions. And this is a peptide that holds high hope, especially because it promotes satiety. And of course, motilin is another one of interest for controlling the gastric and intestinal smooth muscle. Again, you are referred to the animation for more information to appreciate these in greater detail. Now to wrap this up with a short consideration of digestion and absorption. So a villus and a crypt in the small intestine are shown here. So there's endocrine cells in the muscularis, in the deepest portion of the crypts in the small intestine, and that places them deep in the lamina propria near the muscularis mucosa, which separates it from the submucosa. And so you see the lamina propria continues up into the villus, and we see the surface epithelium. See all the microvilli on the surface of those cells, right? They're showing it here, calling it the brush border of the enterocyte. Entero meaning the GI system, the enteric system. And so that's the only barrier between the external environment and the internal environment. And of course the mucus protective secretions that are placed onto the surface. Goblet cells contribute to that, and then there's other glands that will contribute that are differ in a regional basis. There's more of them in the duodenum, getting the contents, the chyme from the stomach, rather than farther down in the ileum. But what's critical here at this point is the arrangement of the neurovascular bundle in the villus. And we see that there is a central lymphatic capillary called a lacteal, and it is surrounded by the arteriolar and venule sides with a very rich capillary bed in between them, and then of course a neural bundle carrying the postganglionics for both of the autonomic divisions and any type of sensory fibers that would be involved in this system. And of course they all join the larger structures located centrally. We see the valves in the lymphatic system to keep unidirectional flow away from the gut wall toward the liver. Excuse me, not toward the liver, toward the lymphatics end up draining into the systemic venous system, as you know. Now let's consider in the lumen nearby what would be happening to the carbohydrates. First, we'll consider this idea of digestion and absorption. So as I said, this is a quick summary of this. You referred to the textbook for additional details. So the carbohydrates are broken down by digestive enzymes into monosaccharides. So the glucose polymers, such as starch and glycogen that we would consume, are acted on first by amylase. This is one of the enzymes secreted in the saliva into the oral cavity. So the carbohydrate digestion will begin with the mechanical digestion in the oral cavity. And these polymers will be digested into the smaller substances, the disaccharides, which are two of the carbohydrate molecules covalently bonded together. And then we have a series of specific digestive enzymes, the maltase, sucrase, and lactase that act on the breakdown products at the disaccharide level. So maltose is two glucoses covalently bonded together, so maltase will generate the glucose from that. Sucrose we're all aware of, so sucrase will break that up into the glucose and the fructose. And then lactose is, of course, a glucose and a galactose. And so if you're lactose intolerant, you don't have an effective lactase enzyme, and you have problems with the lactose affecting things because it doesn't get broken down into the glucose and the galactose. Then the monosaccharides, of course, are available to be absorbed. And in this slide, we're considering the carbohydrate absorption in the small intestine. So we're seeing the lumen here. We're sitting on the top of a villus, right? And we're seeing a capillary here. So this is the vascular capillary, the plasma fluid compartment. And we are following glucose or galactose or fructose from the lumen of the small intestine through a transcellular transport system to the interstitial fluid, where it freely diffuses into the plasma down a concentration gradient and is washed away. So the blood flow maintains this concentration gradient. The apical and basolateral membrane is, of course, the focus of intention. And what we have are apical transporters, and we see the key down here in the bottom right. So this sodium glucose transporter is a symporter. The SGLT symporter is going to transport glucose and sodium into the enteric epithelial cell, the absorptive cell, the intestinal mucosal cell. Not the one of the goblet cells that's secreting mucus, but anyway, there's absorptive cells. Most of them are absorptive cells. So remember, every time we saw sodium being pumped out into the lumen of either the pancreatic duct or the stomach or the duodenum, it's important for the ionic content of the digestive milieu of the lumen, chyme coming in, and then the other terms that apply to it until finally it's feces. But anyway, in addition, it creates this sodium gradient. Again, we see sodium as one of our best friends for creating these potential energy electrochemical gradients to drive other mechanisms. In this case, it's going to pull in the glucose or galactose through the SGLT. At the apical membrane, there is a GLUT5 that is transporting selectively the fructose, or preferably the sucrose. Has a higher affinity for sucrose than the others, but we're showing it selectively here. But at the basolateral membrane, the glucose transporter 2 will be transporting from the cytoplasm to the interstitial fluid both the fructose and the other monosaccharides, glucose and galactose. So all three are being transported by the GLUT2 at the basolateral membrane. So again, it's the selective localization of these to their particular subcellular compartments that's really important. And then again, ATP-dependent transport, the sodium-potassium pump. So you must recapture the sodium that you secreted earlier. And of course, the friend potassium is always around to be cycling in and out of the interstitial fluid and the cytoplasm as a companion to keep all of these mechanisms operating normally. Obviously, if you compromise ATP supplies, like ischemia or hypoxia, then all these processes obviously slow down. Or any electrolyte imbalance is going to affect all of these as well. So continuing on, then we think about the protein digestion. So we start with the the basic peptide structure, the amino acids with the peptide bonds. So peptidases are going to cleave through hydrolytic enzymatic processes these peptide bonds. And see the water here to lyse it, so hydrolytic. And we produce smaller peptides, and then there will be peptides, there's two types of peptidases. Sorry about this. There's those that can target internal peptide bonds, the endopeptidases, and then those that will start at the ends and chew their way toward the center, the exopeptidases. And so there can be aminopeptidases starting at the amino terminal and working their way in. These also are hydrolytic, or at the carboxy end and chew their way in, one amino acid at a time, bump, 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 right? And they may proceed, it may be time dependent or whatever it is. They might only be able to go a certain length into one peptide. There are different factors that affect it, but anyway, they're knocking off an enzyme at a time as they're doing it. So the endopeptidases might be able to produce peptides that can be absorbed as is, or that can then be acted on to be shortened a bit and generate single amino acids by the exopeptidases. So a combination of all these factors generates the digestive products, from proteins down to di and tripeptides that can co-transport with hydrogen. So here again we see the sodium gradient of the lumen driving the apical membrane transporters, and the sodium proton antiporter will generate a local proton gradient that's high in the nearby luminal fluid and low in the cytoplasm. And again, this sodium ultimately is going to get in this cycle here, as we see like this, but some will also be some will also be absorbed through this entire process, as there's net movement in any one direction. Any excess will be taken away to the liver. Anyway, again, it all starts with the sodium gradient. So the sodium that we contributed in the first place is very important. And this drives this local proton pump that'll pull in the di and tripeptides. So it's a proton pump for di and tripeptides. The amino acids, these single building blocks, will be brought in by a sodium amino acid symporter at the apical membrane. And then small peptides are carried intact across the cell by transcytosis, and then dumped on the opposite side. So this is the transcytotic pathway using the cellular packaging, the endocytosis, exocytosis, with the shuttling in between from apical to basolateral membrane. Then finally, the triglycerides are digested into monoglycerides and free fatty acids. So remember, the glycerol backbone and the three fatty acids make a triglyceride. Lipase and colipase are acting on these to split off free fatty acids and leave a monoglyceride. And then the bile salts are going to be contributed from the liver through the gallbladder portion. So the cholecystokinin is going to cause the contraction of the gallbladder and dumping of the bile salts into the duodenum. And they will emulsify the fats as they are generated, the free fatty acids and the monoglycerides, as we saw. Here's the free fatty acids shown here, and the monoglycerides. Some diglycerides become incorporated into this complex as part of the emulsification process. So here we see the bile salt that's coated with lipid droplets. So there's a hydrophobic side that associates with the lipids, and a polar chain side the hydrophilic side that will associate with water. So this is very important for the formation of micelles. And we also see the phospholipids get incorporated into this structure shown in panel B, which are the micelles, the small discs and with bile salts, the phospholipids, the fatty acids, the cholesterol, and the mono and the diglycerides. And so this is what, of course, is going to be absorbed. So here we can see this, and we'll follow it through step by step. So the bile salts from the liver coat the fat droplets. The large fat droplets come from the stomach, and we get the emulsification process that we just looked at. Lipase and colipase will help to degrade these down to smaller structures called micelles. Then the bile salts will be recycled, and here we see the monoglycerides and the fatty acids move out of the micelles and enter the cells by diffusion. The cholesterol is transported into cells by a membrane transporter. So there's a significant amount of cholesterol reabsorption in the small intestine. And then some of the treatments are to absorb this cholesterol and have it pass through in the feces rather than be reabsorbed. So this is dietary cholesterol plus any cholesterol that comes from the bile salts here. And then the absorbed fats combine with cholesterol and proteins in the intestinal cells to form the chylomicron. So in your mind, be associating the micelle with the lumen of the gut tube pre-absorption, and the chylomicron inside the cell post-absorption that contains the triglycerides that were reassembled in the smooth ER, the cholesterol, and the protein. So we break them down so that they can be absorbed, then we reassemble the triglycerides such that they can be packaged and exocytosed to the interstitial fluid compartment, and then the lymphatic system will carry them to the systemic venous blood, and then ultimately that will be delivered to the liver. So remember, the hepatic portal system will take everything except the fats directly from this absorptive process to the liver, but the fats go through the lymphatics, draining into the systemic venous blood first, and then being delivered through the hepatic artery to the liver later. The nucleic acids are digested into nitrogenous bases and monosaccharides, so we digest the DNA and the RNA too. The intestine absorbs the vitamins and the minerals. They don't need any digestion. The fat-soluble ones will be absorbed with the fats that we just saw passively. The water-soluble ones will be absorbed through a transport system. Vitamin B12, when complexed with intrinsic factor, will be absorbed in the ileum. So some people have a deficiency in intrinsic factor and they can't absorb vitamin B12. Vitamin B12 is a very important vitamin that you should investigate. If you are a vegetarian, you need to learn that this is an essential vitamin and for the nervous system and many other functions. And if you don't get the right amount, then it's contained, it's very much enriched in animal products. So you need to take a supplement if you're a vegetarian. It's extremely critical. In individuals that don't have sufficient intrinsic factor, it doesn't matter what they eat, so they need regular injections to bypass the GI system and inject it into the intramuscular in a depot where it's slowly absorbed and directly into the blood. The minerals, of course, are typically captured by active transport. Now we can consider the sodium chloride reabsorption in the small intestine and in the colon. So we've dumped a lot of electrolytes into the lumen we saw that that was critical to form this sodium electrochemical gradient to drive the absorption of most everything. Now we're going through the small intestine and the colon, and we want to capture essentially all of these as much as possible. So we start first here with number 1. The sodium enters cells by the multiple pathways, so there can be an apical channel. It's countered by an efflux of potassium through its leaky channel in the apical membrane. The sodium chloride symporter, bringing both of them in. So again, sodium driving the recovery of the chloride that we saw secreted. Sodium proton antiporter, and the chloride bicarbonate antiporter proteins in the apical membrane will recapture sodium as well as chloride. And then the sodium-potassium ATPase pump that we saw will recapture or reabsorb the sodium and generate the electrochemical gradient to draw the chloride through the basolateral leaky chloride membrane, ion channel. So ultimately we expend energy, so the sodium-potassium ATPase pump to recover the final electrolytes that we had expended energy in some cases to secrete into the GI lumen. In summary then, motility, secretion, digestion, and absorption, and the different regions of the digestive system are shown on this slide. So in the oral cavity and the esophagus, of course, there's swallowing and chewing, and then the saliva from the salivary glands, and there will be lipase contributed there as well. Carbohydrates and fats will be digested, and then there's minimal or really no absorption, although you know that non-nutritive substances can be absorbed through the oral cavity. People put snuff in their lips, and the nicotine is absorbed. Alcohol can be absorbed through the mucosa here as well. But as far as digestive system for capturing nutrients from the external environment, it's essentially none. So we're thinking motility, secretion, digestion, and absorption. So in the stomach, the motility is the peristaltic mixing and propulsion. And then the secretion is the hydrochloric acid from the parietal cells, pepsinogen and gastric lipase from the chief cells. The mucus and the bicarbonate will be secreted by the surface mucus cells. Gastrin from the G cells, and histamine is secreted by the EC cells. Digestion of proteins and fats will occur, and for absorption there will be some lipid-soluble substances, and of course alcohol and aspirin are absorbed here. This is a major site of alcohol dehydrogenase as well, and in males, different ethnic groups have different amounts of alcohol dehydrogenase, but there's also a gender difference where the alcohol dehydrogenase in the stomach is higher in males than in females, and so the amount of alcohol that is absorbed in the stomach directly into the blood is less in males than in females. The small intestine, the mixing and the propulsion are primarily by segmentation, so not as much peristalsis as we said for the comparison with the esophagus. The secretion will be enzymes, of course, the bicarbonate and the enzymes from the pancreas, the bile from the liver, mucus from the goblet cells that we saw, hormones, the CCK, secretin, GIP, and other hormones, referring you again to the table. Complex signaling system that challenges us eternally, it seems, but major discoveries are being made all the time to help us understand this system much better. The carbohydrates, the fats, polypeptides, and nucleic acids are all going to be digested to one degree or another here. And then absorption, of course, the peptides by the active transport that we saw, the amino acids, glucose and fructose by secondary active transport, fats by simple diffusion, water by osmosis of the ionic movements, the minerals and the vitamins by active transport. Then in the large intestine, the motility is a segmental mixing, the mass movement for propulsion. The mucus will be secreted by the goblet cells to protect the gut wall. There's really no digestion. As bacteria will perform some here, but nothing contributed by the organism itself. Absorption of ions, water, minerals, vitamins, and small organic molecules that may have been produced by the bacteria will occur here. And of course, there's a summarization and additional detail provided to the